Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and welcome to the live session 2 for the course Basics of Biology. So if you have any questions, you should post your questions so that we should initiate the discussion and we should be able to, I should be able to clarify your doubts. Hello everyone, this is Dr. Vishal Trivedi, Department of Biosciences and Bioengineering, IIT Guwahati, and welcome to the live session 2 for the course Basics of Biology. Yes, so just shift to the PT. Yeah, so I think Sudarshan is asking about the differences between the cell-mediated immunity versus the humoral immunity. Okay, so let me give you a brief idea what is cell-mediated immunity. So if we have the immunity, right? When you talk about the immunity, immunity has two components. One is the humoral immunity, and the other one is called as the cell mediated immunity. Okay, so very simple way of understanding the what are the components are going to fall under the humoral immunity is all the things which are going to be secreted and be present as a liquid in the particular microenvironment that will contribute for the humoral immunity. Whereas the cell mediated immunity means here the cell is actually going to participate directly into the reaction. Okay. Let me give you an example. Okay, example is that for example the antibody is a part of the humoral immunity, but the B cell or even T cells are the part of the cell mediated immunity. because this, when there will be a pathogen, for example, if you have a bacteria, right, and if you have the B cell, right, the B cell is directly going to interact with this particular bacteria, and it is actually going to engulf, right, and that's how it is actually going to present the the antigenic peptides from the bacteria along with the MSC class 2, and that's how it is actually going to stimulate the the T cell. Okay, and then what will happen is, Okay, sir. Okay, so this is what I think we were discussing. Sorry about this technical issue, because there was some issue with the displaying of the PowerPoint. Okay, so that is what we were discussing. We were discussing about the immunity. So immunity has two components. One is called as the humoral component, and the other is called as the cell mediated component. Humoral component, you can easily understand what is being a part of the humoral component is whatever the liquid is going to be secreted by the cell is actually going to contribute for the humoral component. For example, the antibodies. Antibodies are actually going to be a part of humoral immunity, whereas the B cell, T cell, any kind of cell, whether it is macrophages, all these are actually going to be a part of cell mediated immunity, because here directly the cell is going to involve. Okay, that does not mean that the cell is not going to be involved in the humoral response also. You know that the antibodies are actually going to be secreted by the B cell only. Okay, so this kind of classification is only being done so that we can be able to understand the immunology, we can understand the processes much simpler way. Okay, but as such, when you see the immunity, it is actually a mixture of both the components. Okay, so apart from this, Sudesh is asking about the necrosis. Okay, so what is necrosis? Okay, so I think he has also asked about what is the difference between the apoptosis and the necrosis as well. Okay, so when a cell is actually going to go for a death pathway, okay, it has two choices. One, it can actually go for the apoptosis. The other is can go for the necrosis. Okay, so now if you want to understand the major difference is that the apoptosis is a safe mode of death. Okay, whereas this is actually going to be not safe for the organism. Why it is not safe? Because in the case of apoptosis, there will be no leakage of the cellular content, whereas in the case of necrosis, it is actually a leakage of the cellular content. Now, necrosis means cell is actually going to be burst. Okay, so necrosis is not a programmed death. It means necrosis can happen all of a sudden, and that's why one of the classical example is that necrosis will lead to the cell burst, and the cell burst is actually going to be responsible for the leakage of cellular content, and when there will be a leakage of cellular content, it is actually going to be responsible for inflammation. Okay, and that inflammation is going to cause the tissue injury. And that's why I said that the necrosis is not a safe mode of death for the organism. Whereas in the case of apoptosis is a programmed death. Okay, so there is a well-defined steps what is going to be followed by the cellular pathway. You know that the apoptosis could have the two different pathway. It could be either the death receptor pathway or the mitochondrial pathway, right? And or it could be the mitochondrial pathway. And then ultimately both of these pathways are actually going to activate the caspases, and the caspases are actually going to activate the DNases, and the DNase is actually going to degrade the DNA, and that's how it is actually going to cause the death. Now, since the apoptosis is a programmed cell death, it will lead to the generation of the apoptosome. Okay, and the apoptosome is apoptosome means that if there is a cell, right, and if this cell will go for the apoptosis, it will actually going to be get broken into the multiple small apoptosome. So that's why this cell is actually not going to release the cellular content. All these individual apoptosome are actually going to be engulfed by the macrophages, and that's how there will be no inflammation in the case of apoptosis, and that's why the apoptosis is considered to be a safe mode of death. Now, Sudesh is again asking about the one more aspect. One is does our immune system attack the tumor cells? Definitely, our immune system attacks the tumor cells. Okay, the cytotoxic T lymphocytes and the target. The tumor cells are the target of these cells, and the process is remains same whether it is a tumor cell or whether it is a bacteria, except that the final executory T cell is going to be different. In the case of bacteria, it is going to be T helper cell, whereas in the case of the tumor, it is going to be TC cell. Okay, so but the process remains the same. So immune system do attack the tumor cells, but since the tumor cells are our own cell, sometimes they actually have the mechanism so that they can be able to escape the attack mechanism, and that's why it is when that happens, those cells are actually going to be grow and cause the development of the tumor. I'm sure, Sudesh, you will be satisfied with the answer. If you are not, you can post me some more questions, and probably will be happy to answer. This slide is not changing. I think you have to change the slide, right? No, in the display it is not. The YouTube display, it is still showing the humoral immunity, that old one. Now it's showing my face.
Change it to the slide.
जब मैं लिख रहा था तब स्लाइड पता नहीं दिखा रहा था कि नहीं. ओके, तब if you have any more queries, it's better you ask me right now, and you can, since this live session is not very long, you should take the full opportunity of this to ask me any questions or clear your doubts. so that it will help you to prepare for your exam next month. So Darshan is asking about the minimum passing marks for the exam. So I think the minimum passing marks is 40, which includes the uh, marks from your exam and marks from your assignments. But you are supposed to be clear the assignment and as well as the exam. So there is a minimum for both, and then only you will get the certificate. Um, Siddesh is asking about when he will get the hall ticket. I really don't know when we're going to get the hall ticket, but you can contact the NPTEL team at IIT Madras or at IIT Guwahati, and they'll be happy to help you. And if you have a query related to the exam center, I think in most of the major cities, the exam centers is going to be there. So you don't have to worry about the exam center. It is going to be close to your place. If you have any queries, you should post your questions so that we will have a discussion about that, and I hope that will help you to face the exam next month. This session is very small, so I think you should take the full opportunity, and you should exploit as much as possible to interact with me and clear your doubts. Okay, I think Sudarshan is asking about what is phase and what is trans phase. Okay, so I'm sure for this I might have to draw a cell. Okay, so what you see, you have a cell, right? Then you have the nucleus, right? So this is the plasma membrane. This is the nucleus, right? And around the nucleus, you have the endoplasmic reticulum, right? Don't worry about this drawing because drawing is going to be bad, right? And then we have the Golgi bodies, right? Right? So the phase, so you're asking which one is the cis phase and which one is the trans phase. Okay? So the phase through which, so this is the cis phase, this is the trans phase. Okay? So the molecules are actually going to enter through the cis phase, and you know that the molecules will enter through the cis phase and they will exit through the trans phase, and in between you have the medial Golgi also. Okay, so and you know that the Golgi bodies receive the material from the ER, right? So this is your ER. Okay, so the phase which is actually facing towards the ER is going to be called as cis phase, and the other side is going to be called as trans phase. Okay, so I'm sure this is clear, right? So the phase which is towards the endoplasmic reticulum is called as cis phase. The phase which is towards away from the endoplasmic reticulum is called as trans phase. Okay, I'm sorry about this drawing because the drawing is not that good. But if you see the drawing in my lecture notes, probably it would be not an issue. And I hope it's clear, right? In fact, in my lecture note also it is written that which side is cis and which side is trans actually. I hope it is clear to you, Sudarshan. शेयर कर दो ना.
Which issue are colors?
पहला स्लाइड ही दिखा रहा है. Okay, it's okay. Okay. So Siddesh has another question, and that is very interesting question actually. That lysosomes are having the acidic pH, okay, and it has very various enzymes also. So how does the plasma membrane withstand such a low temperature, low pH? Okay. So now you understand one thing, okay? Plasma membrane is mainly being made up of lipid, okay, and it is made up of the protein. So protein is a minor fraction, whereas the lipid is the major fraction, okay. Now protein is hydrophobic in nature, right? And that's how the lipid molecules are sticking to each other, and that's how they are actually giving them the continuous, right? You know that the heads are actually making with each other, right? And that's how you have the lysosome. So this is the lysosome, right, where the lipids are actually mixed with each other, and that's how you have the uh, the chains in the center, right? Now, if you have the acidic pH, right, the acidic pH probably may affect the protein component, but it will not going to affect the lipids, because what it is going to do, if you have a low pH, that only going to change the ionization states of the polar molecules. It is not going to change the ionization status of the non-polar amino acids, like non-polar substances like and you know that the lipids are non-polar, so it is actually not going to affect the integrity or the interaction between the lipid molecule, because if you change the pH, nothing will change or nothing will actually going to affect the assembly of the lipid molecule, right? The assembly of the lipid molecule will remain intact. If you add, like for example, if you add the acetone, or if you add any other of the hydrophobic substances like hexane, then only it is actually going to affect the integrity of the plasma membrane. Changing the pH or changing that kind of situation is not going to affect the integrity of the plasma membrane. Forget about the lysosomes. Think about your own stomach, right? When you take the food, right, and it get insert when you take the food, right, the pH in our stomach is it goes as low as around pH 2, and lysosomes are also having the similar kind of pH level, right? But nothing happens to our stomach, because the stomach is nothing will happen to the cells also, because the same reason that it does not affect the cells to a lot, because at least it will not going to harm the plasma membrane, because the plasma membrane is made up of the lipids, and lipids are more sensitive for the hydrophobic substances or solvents rather than the hydrophilic. I hope it is clear, Sudesh. If it's not, we'll again going to discuss some more.
आप मेरा चेहरा दिखा लीजिए, खत्म करिए।
I request you all, you should post your questions so that we will have a fruitful discussion about that particular topic, and it may help you for preparing for your final exams. Because if you don't post the questions, probably we'll terminate or we'll going to terminate the live sessions. It seems we don't have any more questions, so I'm thankful to you for joining the live session 2, and I hope we will be having a fruitful discussion in the live session 3. So with this, I would like to conclude this live sessions. See you in the live session 3. Thank you.